Hi children, in today's session, let us continue with the types of cells. In previous session, I already specified with the eukaryotic cell taking example bacteria. Moving on to the eukaryotic cell, standard examples taken as plant cell and animal cell. Let us discuss one by one, followed by the differences between plant cell and animal cell. Remember, whatever we are studying today, it is the most important part for exam point of view. You have to practice the diagrams given in the textbooks. Plant cell and animal cell with minimum 10 labelings will attract 5 marks each. Any one diagram is compulsory, followed by differences between plant and animal cell also carry 5 marks weightage. So, in today's session, we are dealing with total 3 answers. Coming to the plant cell, it is either meristematic, parenchyma, collenchyma, sclerenchyma, a component of xylem or a phloem, whatever it is. Standard example considering all the plant cells is shown as a diagrammatic representation. It is polygonal in shape, bounded by cell wall, which is discontinuous layer having plasmodesmata. Now these plasmodesmata are cytoplasmic strands which connects one cell to the other for the transport. This cell wall is composed of cellulose. It is thin in condition, known as primary cell wall. For extra mechanical support, this cellulose is deposited with hemicellulose or pectin or lignin or cutin or suberin, etc. Then the cell wall becomes secondary. Between the two cells, there is a cementing substance composed of magnesium pectate or calcium pectate. This we call middle lamella. Just imagine the wall constructed. There are bricks and the concrete. Now, between the bricks, whatever the cementing layer comes, it is for the support and extra mechanical strength. Similar way, when the cells adhere to each other, they will have this cementing substance known as middle lamella. Coming inside, there is a cell membrane, plasma membrane or plasma lemma. It is composed of phospholipids, proteins and oligosaccharides. It is said to be semi-permeable in nature, allowing only few components to pass through, while cell wall is said to be fully permeable layer. Inside the cell membrane, we can see the cytoplasm with peripheral nucleus small in size, as in functional plant cell, maximum space occupied by vacuole. Vacuole is a storage organelle. Whatever food is prepared in photosynthesis, it is stored within the vacuole. The unique membrane around the vacuole is called tonoplast. The nucleus is covered by nuclear membrane having minute openings called as nuclear pores. Inside the nucleus, there is nucleoplasm having chromatin network. Each chromatin fiber becomes short and thick. At the time of cell division, we call it as chromosomes. Chromosomes are composed of proteins and the DNA, and DNA is a genetic material. Hence, this nucleus is said to be a master center within the cell. Apart from vacuole, there is endoplasmic reticulum extended from nuclear membrane to cell membrane. It is storing lots of biomolecules within. On the surface of endoplasmic reticulum, there are minute ribosomes. These are the sites for protein synthesis. There is a powerhouse suspended in the cytoplasm called mitochondria. There are functional plastids. Plastids with chlorophyll we call chloroplast, with accessory pigments known as chromoplast, and the plastid storing biomolecules known as leucoplasts. Apart from this, there is a Golgi complex. It is a packaging unit in the cell which receives the raw forms of biomolecules which are processed and packed within the vesicles for the transport. there is a lysosome. It is considered as suicidal bag. In the lysosome, hydrolytic enzymes are present which will digest unwanted component generated within the cell. So, plant cell has 7 cell organelles like endoplasmic reticulum, Golgi complex, mitochondria, plastids, lysosomes, vacuoles and ribosomes. Let us come to the animal cell. It is not in a fixed size and shape. It is covered by cell membrane or plasma membrane. Cell wall is completely absent. There are minute extensions we can see on the surfaces known as microvilli. Inside the animal cell, there is a cytoplasm with the nucleus present in the center, as usual, protected by nuclear membrane with chromatin network, and there is a nucleolus both in plant cell and animal cell located inside the nucleus. This nucleolus is synthesizing center for DNA and RNA. Apart from the nucleus, in the animal cell there is a powerhouse mitochondria. There is Golgi complex. There is endoplasmic reticulum extending from nuclear membrane to cell membrane, smooth without ribosomes, rough, having the ribosomes on the surface. Lysosomes are more in number. In animal cell, being a heterotrophic in nature, no plastids are found, no vacuoles are found. So when we compare plant cell with the animal cell, there is one more extra cell organelle present in the animal cell known as centrosome. This centrosome is having centrosphere and perpendicularly placed a pair of centrioles which will produce spindles at the time of mitosis and meiosis to facilitate the chromosomal movements. Now, let us compare plant cell with the animal cell. Plant cell's shape and size is fixed. Animal cell not. Plant cell covered by cell wall with plasmodesmata. Animal cell without cell wall. Plant cell having 7 cell organelles, peculiarly plastids and the vacuoles, while in animal cells 5 cell organelles, devoid of plastids and the vacuole. Centrosome is absent in the plant cell. It is present in the animal cell. With this, I have completed today's session on the introduction to eukaryotic cells, taking example plant cell and animal cell. In the next few sessions, we are dealing with each component of the cell. So this forms the topic cell, a basic unit of life. Thank you very much.